Okay, hi grade 10s. What I'm going to do over the next couple of lessons is just go through the different parts of the cell. So it's one PowerPoint, but I'm not going to do one video on the whole PowerPoint. That would just take too long. So we're going to do different parts of the cell, bit by bit by bit. Okay, so just open up here. Okay, so this is the PowerPoint called Cell Structures. All right, so what we have here are something that we should be familiar with by now. So these are two electron micrographs. This one's an animal cell and that one's a plant cell. And you should be able to see why, because the plant cell is surrounded by a cellular cell wall. You can see the nucleus nice and clear, nucleolus, and there's the DNA. Now, if you remember, DNA is thread-like, okay? So lots of long little threads, think of wool, but because you're cutting through the threads, you're going to see like the point of my pencil. And that's why you see lots of little dots. That's the DNA in the nucleus. These are the chloroplasts, these big long things. And in between here are the tiny little mitochondria. There's a large vacuole, and the gray mottled area, that would be the cytoplasm. All right. And you can see how two cells are joined. Now that region there we're going to talk about later. That's called the middle lamella. It's like it's a substance which glues the two cells together. Okay, then here you can see an animal cell that they've added color to. So there's the nucleus, but we can't see the nucleolus because maybe it's behind or in front of the view that I'm looking at here. And these little granules here, we'd need a higher magnification, but I'm guessing that those are mitochondria. But we can't really see much more detail here. We would need to go or look at a higher magnification. So now we've looked at, we've calculated sizes of cells, and we're going to finish that at a later stage. So remember, we looked at molecules at the beginning of the year, lipids, proteins, etc. So now, those can be viewed with an electron microscope, but a light microscope, you can see a chloroplast. You can see the oval or round-shaped chloroplast. You just won't be able to see the detail of the chloroplast, and that's what you would need an electron microscope for. But you see chloroplast in the cell? Yes, you can with a light microscope. Bacteria, you can see little round shapes or oval shapes, etc., with a light microscope, but more detail with an electron microscope. And then plant cells, you can see here, are between 10 to 100 micrometers. So if you remember, 1 millimeter is 1,000, 1 millimeter is 1,000 micrometers. So 10 cells would be able to fit next to each other in a millimeter on your ruler. All right, and there you go, what we can see with the unaided eye. Okay, so we have looked at examples of this before. So here we have cells of different sizes, and you can see they're different shapes as well. Animal cells are not only rectangular. All right, so red blood cells, only 7.5 micrometers. White blood cells are bigger. Smooth muscle cells, like the muscle cells in your esophagus, they are long and spindle shaped and 200 micrometers. So 5 of those would fit lengthways in a millimeter on your ruler, a millimeter being 1,000 micrometers. And you can see here a human egg cell is 140 micrometers, so maybe about 8 of them would fit along a millimeter in your ruler, maybe less. All right, so this relates to the diagram that I showed you yesterday, which I drew, and then I showed you what was common and what was different in plant and animal cells. But now we've also got what's the difference between prokaryotes and eukaryotes. So remember, prokaryote cells, like bacteria, do not have a proper nucleus, but they would have a cell membrane. They even have cell walls, all right, of different substances. They have DNA, and the DNA is suspended in the cytoplasm. So there is no central nucleus. So pro means simple, karyote refers to the central part of the cell, the nucleus. So they don't have that. The DNA is suspended in the cytoplasm. But you guys, we have got cells that are eukaryotic, so it's more advanced. There's a nucleus, all right? So eukaryotic cells have got membrane-bound structures. So all the organelles, okay, they're not found in bacteria, nucleus, ER, Golgi body or apparatus, little lysosomes in animal cells, vacuoles, mitochondria, and they even have that cytoskeleton, those crisscrossing network of fibers which give the cell cytoplasm support. All right, then animal cells and plant cells are both surrounded by a cell membrane. They both have ribosomes, nucleus, ER, Golgi, mitochondria, cytoskeleton, cytoplasm. All right, so it's very similar to the previous list, but animal cells only have centrioles, lysosomes, and small vacuoles in some, like amoeba, because they live in a watery pond, whereas plant cells have cell walls, chloroplasts, and large vacuoles. Little vacuoles join to become big ones, and those are in plant cells only. So this is a nice Venn diagram to show, to compare the different cells. All right, then we look at bacteria, which are prokaryotes in grade 11. They can be rod-shaped, they can be spiral-shaped. I'm looking for one. Okay, I'm not seeing it here. Some of them can have flagellae, some of them can be round, large, small, in clusters, etc. These are little round ones, and you can see they're dividing by mitosis, asexual reproduction. So they are, if you look in the middle, they have the DNA in a region that's called the nucleoid. So there's no nuclear membrane. There's ribosomes in the cytoplasm, so they can make protein. They have a cell membrane, so that's a plasma membrane, all right? Edge of the cytoplasm, just like other cells. And then there are walls. There are bacterial cell walls made of substances like a protein sugar, peptidoglycan, etc., or a mucus layer. And there's a capsule which enables, it's like cell recognition. Okay. Then, if we look at cell membranes in general, okay, so think of a cell and it's around, the edge of it is more organized. It's not really a separate layer. It's more organized. We call it the cell membrane, and it's got to control what moves into and out of the cell. So we say it is semi or differentially or even partially permeable. It only allows certain molecules to pass through. So I've just got a piece of paper here and a pointy thing, and all I'm doing is I'm poking holes in my piece of paper, and I'm trying to represent the little pores in a cell membrane. Maybe you can see those pores. All right, so those pores only allow small molecules to enter or leave the cell. So that's why we say a cell membrane is differentially or semi or partially permeable. Only certain things can move across the cell membrane, because you don't want everything just to leak out. All right, that cell membrane is the edge of the cytoplasm, like when you make custard or porridge and the top part, in the pot, and then the top part becomes a little bit harder, like a skin. Well, that's cytoplasm. It's a boundary. It's made of molecules, and we'll talk about those. So if this diagram over here, there's part of one cell that they've colored in, there's part of another one. There's the cell membrane of cell 2, and the other blue line that's been color added is the cell membrane of cell 1. And there's a little space between the two cells, and that's called the intermembrane space. And that's kind of where they interact with each other and where molecules move around. We'll see later. All right, now, what are cell membranes made of? They're made of proteins and phospholipids. So you might remember that from term 1. And sugars, like carbohydrates like sugars and other molecules. Okay, so small molecules need to move across. Now in grade 9, we talked about how water moves across or into a cell or out of a cell by osmosis. It moves from high concentration through the membrane to low concentration, simply by moving from where there's more of it to where there's less of it. Sorry. Okay, and salts, sugars, oxygen, CO2, they would diffuse into the cell. So osmosis is diffusion of water, but we say that all other molecules move by diffusion from where they are more in concentration to where they are less in concentration. And later on, we will look at something called active transport, where the cell uses energy to move molecules in or out. All right, so membranes, they not only do they surround the cell, but organelles are separated from the rest of the cell by membranes. So chloroplast membranes, nuclear membranes, mitochondrial membranes. The cell is now compartmentalized into different regions which have specific roles. Okay, so bacteria, prokaryotic cells would not have those organelles, but eukaryotic cells, there's now separate compartments where different functions of the cell take place. Now, synonyms for cell membrane, it's plasma lemma. Lemma means like a layer, or plasma membrane. Plasma referring to the cytoplasm, edge of the cytoplasm. Now, this is what a cell membrane looks like. So, it's made up of a number of molecules arranged in a specific way. All right, it's made up of two layers. So, that's why it's called a bilayer. Now, if you remember from term 1, those little phospholipids, my hand is the phosphate part, okay? And then you've got the two little fatty acid chains. Well, glycerol and the phosphate, and then the two little fatty acid chains, that's what phospholipids are. Okay, now if you look at this little model that one of the grade 10s made in a previous year, let's maybe just turn it around, and I've got some over here. These are the little phospholipids. All right, so there's the little glycerol. There would be a phosphate group attached here and the two fatty acid chains, and they line up in two layers facing each other. Okay, so that's why we call a cell membrane a lipid bilayer. It's made up of two layers of lipid, and these blue things are the proteins. And the proteins help to transport substances across the cell membrane. So they act as channels. So if you look on this side where one is cut in half, it's like a little gate, like a little entry and exit point through the cell membrane. All right. Now, if we look at the cell membrane, it's got lots of other little things. These little beads, those are the sugars. So they attach to the protein molecules, and they are like the aerial saying, hey, I'm a muscle cell. You come here, okay, depending on what the muscle cell needs. So those are the receptors, the sugar molecules attached to proteins or anywhere else, okay? So if you look at this diagram, there is the lipid bilayer, all right? There's the glycerol phosphate groups, and here are the fatty acid chains facing inwards, and there's a reason for that. And then you've got these proteins. Now, either they pass all the way through with a channel in the middle, so they're called a channel protein, or you get proteins that are on the exterior or interior surface, or they go halfway through or all the way through, etc. So there's different kinds of proteins. Now, if we look here, in between these little red diamonds, those are cholesterol. So yes, we do need cholesterol in our body because it helps to make our cell membrane. So on this model, that little beads over there, that's the cholesterol molecule. And they basically, they enable those phospholipids to not be too close to one another. So the cholesterol would be in between saying, guys, space out. So cell membranes, the molecules tend to move apart on hot days, or they tend to go together on cold days. So the cholesterol molecules in between are saying, no, 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 not too close, not too far. They're making sure that the distance between the phospholipid molecules stays constant. Okay? Like when you stand in a row and you put your hands out and you make sure that the next person isn't touching you. Like when you line up in marching in the army, I suppose. All right, so there are pores in the membrane, like through there. Okay, and molecules can pass through the membrane itself as well. All right, now we say that, okay, it's a lipid bilayer, and we talk about the fluid mosaic model of a cell membrane. Okay, so fluid refers to the fact that these little molecules here, they can all move around. So they can change position. Okay, the proteins can move and everything can move around, like having lots of marbles on a tray, and then the larger ones representing the proteins, and they can move around. They're not static. They don't stay in one place. So that's why it's the fluid mosaic model of a cell membrane. Mosaic refers to the patchiness. All right, and I'll show you that on another slide. And then obviously bilayer, because there are two layers of lipid. So let's just go through the labels here. Now, the reason why I know that's the outside of the cell is because there are those little sugar areoles saying, I'm a muscle cell, you need to come here, whatever the cell needs. So that's the outside of the cell. And then at the bottom would be inside the cell. Here would be the cytoplasm of the cell. Okay, then There's a membrane protein, there's the cholesterol, making sure that the phospholipids aren't too close or far from one another. The carrier protein with the channel in the middle. There's sugars, carbohydrates attached. There's the phospholipid bilayer. The cell cytoskeleton, if you remember those fibers through the cell cytoplasm, they organelles, can move along, and it gives support to the cytoplasm as well. All right, now this is a mosaic. I like doing mosaics. I haven't done them for quite a while now. But yeah, it's patchiness. So you've got different molecules in between each other, just like the sugars, the cholesterol, the lipids, and the proteins forming the cell membrane. Okay, there's a lovely stereogram, three-dimensional diagram of a cell membrane, and nice the way they pulled it apart here. You can see the two layers of phospholipids. You can see the cholesterol in between, keeping those phospholipids at an equal distance from each other. You can see the proteins, some of them going all the way through, some of them with channels, some of them on the outside, some of them halfway through, like here as well. And you've got the sugar molecules attached to some of those proteins saying, I'm a muscle cell, come here. Now, these two words, hydrophilic and hydrophobic, those phospholipids, the reason why they line up like that, okay, is because, all right, the fatty acid chains don't like water. We know fat doesn't like water. So they line up facing one another. So they are hydrophobic. But the head, the glycerol phosphate groups, they don't mind being next to the outside of the cell. And in between cells, there's a little bit of tissue fluid, which is watery. And they don't mind being next to the cytoplasm of the cell. So they face the watery part, whereas the fatty acid chains, they face each other because they don't like being, they're hydrophobic, they don't like being near water, whereas the head parts are hydrophilic. They don't mind being near watery tissue fluid outside the cell or cytoplasm inside the cell. All right, and I will upload the videos for you to have a look, and we're going to draw a diagram later. And we will have a look at some animation. Now, I'm not going to do diffusion, osmosis, and active transport here. I'm actually going to keep that as the last lesson on cells, just because it makes more sense to do it there. All right, that's part number 1 of